I'm Ira Herskowitz, and I've been a professor at UCSF for now 21 years. And before that, I was on the faculty at the University of Oregon for 9 years.
So let's begin by having you tell us a little bit about what led you to be interested in science.
Well, my first interest in science, I think, was probably when I was in grade school in Bloomington, Indiana. My father was a Drosophila geneticist, and he worked in Muller's lab, H.J. Muller. And my brother and I, my twin brother and I, used to go to the lab, and we didn't do anything like do crosses or anything like that. We took these little counters like this, and we had contests to see who could do the highest number. We had speed contests, at any rate. One thing they had in the zoology building in Bloomington called Jordan Hall, they had the kind of displays that you never look at, and you assume nobody ever else looks at. And this was a display of pickled fetuses, human fetuses, and it blew me away. And I was very intrigued by these fetuses. And then when I was in junior high school, I was 13, my mother was pregnant, and she said, she was musing over how amazing it is that a fertilized egg can give rise to an embryo with many different kinds of cells, and how amazing that was. And so those things got me interested in developmental biology.
When did you first consider science and biology as a career option?
Well, I came across the Microbe Hunters by Paul de Kruif. Have you read that?
Oh, yeah.
Uh-huh. And I remember the chapters about Louis Pasteur, and just I think that book really exposed me for the first time to scientific discovery. And amazingly enough, of course, I did turn out to be a microbiologist, so there's a connection there. I tried to read books about physics, and of course I understood none of it. I also read a lot of books about the Brooklyn Dodgers, my favorite team, and about mountain climbing. But I was a good student, I enjoyed things, and I think I sort of always had the benefit of very good teachers.
Which of your teachers in high school or junior high stand out in your memory as being particularly influential?
Well, one teacher that was very influential was my 7th grade English teacher. We had just moved from Bloomington, Indiana to University City, Missouri, and I wrote a report in this English class on New York City. It was something like 13-page single-space type. I wanted to blow him away because I was new in town. And it consisted of something like the first library in New York City was in 1789, and the first disc was 1784, and it was horrible, and I got a C. And so I was immediately exposed to teachers who had standards, and I really sort of shaped up there, and I learned, that was the beginning of learning how to write. And then in high school, there were a series of exceptional teachers, a math teacher named Miss Bruns, and I really did very well in math. I liked it, and I actually thought I might be a math major. A very, very good English teacher named Mrs. Gottlieb, who one of her students just won the Pulitzer Prize this year in poetry, so we're all very excited about that. And my science teaching in high school was between bad and pathetic. The biology teacher was horrendous, the chemistry teacher was confusing, and the physics teacher was useless.
Could you tell us a little bit about what took you to Caltech as an undergraduate?
Well, my father, being a geneticist, knew that Caltech, which everybody would think of as an engineering school, was very strong in genetics, and of course Caltech was. And so that was recommended as one place to go. I was clearly interested in science. But I went to Caltech as a math major, and I placed out at the first year of math, which was an incredibly stupid thing to do. Got into a class where I got a 0 on my first 3 exams. So I started out Caltech. It was quite a shock. It took me, 10, 15 years to recover. I forgot to say that I also, on my chemistry midterm, got a 3 out of 125. So it was, wow. It was quite something. But the really good things about Caltech were clearly that I began working in a lab as a junior, worked in Bob Edgar's lab. And the courses that I had at Caltech, especially some genetics courses, were spectacular, and I got turned on to genetics. Actually, But you take anybody of any ability and you can always give them more than they can handle. And that was the sort of exactly the mode in which I existed for 4 years at Caltech. And it was only after leaving Caltech, when I was a graduate student at MIT, that I realized I had learned a huge amount at Caltech. Because when I got to MIT, I hit the paths running.
What was it like being a beginning undergraduate in Edgar's lab, which I assume must have had some fairly talented and experienced graduate students and postdocs at the time?
The Edgar lab was just amazing. The really hot stuff in biology, in the new molecular biology, was phage. And he worked on T4, and he was one of the people, along with Epstein, who developed conditional lethal mutations. And so as I got into the lab, there were incredibly good graduate students there. The person I worked with was Jonathan King, now professor at MIT. And Jonathan was not only a wonderful mentor, but a very dear friend. And I can remember vividly Jonathan showing me in the first day of the lab, I got in the lab, how to pipette, and what mistakes that you would make and not to make, and things like this. So he was fantastic. Sandy Parkinson was a graduate student there, and we were friends, have been friends for many years. And then Bob Husky and Dick Russell, and it's a very, very good group of students. They were totally into it. They loved it. They lived and breathed phage, and it was very stimulating for me.
Now, Sandy Parkinson gave me reason to believe that you might have actually gotten out of the lab on perhaps a camping trip a time or two.
Well, of course, the Edgar lab was right next to the Delbruck lab, which was famous for camping trips. And to be quite honest, Jasper, about getting off campus, in my whole freshman year, I got off campus 0 times. Caltech's campus at the time was 3 block by 1 block. I never left it once. In my second year, one of the first things I did when I came back to in September, this was 1964, I went to see Bob Dylan sing at Long Beach High School. Then I got back to Caltech to tell everybody about this great concert. Nobody there knew who Bob Dylan was. At any rate, yes, I did go on, I think, 1 or 2 camping trips to Joshua Tree, and it sure made me wish, in retrospect, I'd done more. It was beautiful scenery and wonderful people, and I didn't do enough of that.
What was your first experiment?
I worked on auxotrophic mutants of T4, which seems strange. That was an idea Delbruck had. But it was cofactor acquiring mutants of T4. Mutants of T4 that required, in this case, tryptophan in order to absorb. They had been known. Somehow these phages would grow, and in this case absorb only in the presence of tryptophan. And so my project was to isolate more mutants of that kind, and then to try to find out what genes these mutations were by doing various complementation tests.
You described the first year as being academically a bit of a hard haul. Did you have doubts that you would actually be able to get through at any time?
I had doubts at all times. So let me tell you some of the things that happened when I was working in the Edgar lab that really influenced me, and other things that happened at Caltech that made it clear that I wanted to be a geneticist. So first of all, I remember the Edgar lab was working on morphogenesis, and morphogenesis was the heads and the late genes, the head genes, the tail genes, the base plate, and all that business. And then I just remember thinking, DNA replication occurs before morphogenesis. So they're early genes and they're late genes. What is it that makes that order? Because the DNA is always in the cell, but these genes are expressed and these aren't. Why is that? So that really grabbed my attention. And early on, I was very interested in gene regulation, and that really hit home.
Were the people in the Edgar lab focused on regulation? Or
Not
was
at
it
all,
no.
no. None of them worked on regulation. And I didn't either.
Yeah.
I remember one group meeting presentation, I think it must have been a journal club. I was in my senior year, I think, was on the newly discovered lac repressor. And it was, I think, a preprint or something, and it was work of Mullerhill and Gilbert. and how they had used radioactive IPTG to find an IPTG binding protein, and I loved all of that stuff. I mean, that is exactly what I wanted to be doing. I was absolutely intrigued by that. So that had a big impact on me, more than morphogenesis. Another moment that, I think there are two genetic things that really stand out in my mind as an undergraduate at Caltech. One is, they're really the same. It's the relationship of gene and protein. I remember sitting in a class by Norman Horowitz, but he had Bruce Ames come and give some lectures. And Bruce Ames was trying to figure out the regulation of the histidine the genes involved in a histidine biosynthetic operon. We went through the experiments about trying to isolate mutants that were constitutive or uninducible, and that really I found absolutely fascinating. And then also, I remember hearing about amino acid analogs that caused false feedback inhibition. They would bind to the first enzyme in the pathway and then shut down synthesis of histidine, and then the cell would starve for histidine. So then what was a great moment was people isolated mutants that were resistant to feedback inhibition, and then they mapped them, and they mapped to a particular place in the gene. And I could really see the relationship between the function of the protein and the gene, and how you could use genes to study proteins. You isolate mutants, you learn about proteins, and also because some information could be gleaned biochemically about proteins, you could use proteins to learn about genes. So that was burned right into my brain cells. And then, of course, in the Edgar lab, they used TS mutants and they used nonsense mutants. And I could really see the connection of those mutations and protein function. So I think very early on, I was very comfortable with genetics, but I always related it to proteins. And I think that's something that I carried through with my graduate work and then future work.
So with your father being a noted geneticist and you turning towards genetics, I wonder to what extent there's a relationship there.
I've thought about that too. I mean, in what way does having a father who's a geneticist influence me? Well, first of all, I knew that it was a profession like bus driver where you could earn a living. It's not like there's anything fascinating or romantic about it, or he seduced me into working in the lab. He never once suggested, Ira, why don't you come to the lab and push a few flies around. So there's nothing directly there. But again, as I mentioned before, he rooted me towards Caltech, and not many people would have known to do that, a place that's so strong historically in genetics. So I think those are the two most obvious ways. It's nothing that direct, but there is something very nice about having a father who's a geneticist, which is that my father's 82, and he loves talking about genetics with me. He loves following the work going on in my lab, and he's my biggest fan, really, in that vein.
When you were in high school, did you talk to him about what was going on in his lab?
No. My father did a master's degree with L.J. Stadler, and then later, after Stadler died, my father said, this is really unfortunate that these great geneticists had never been filmed or recorded. And so that set him to develop he was approached to do a TV series on genetics, and he immediately leapt to do it. And he got people like Muller and Ed Lewis and Ralph Cleland and Tracy Sonneborn, Jim Watson, Josh Letterberg, Kurt Stern, a variety of people to participate. And I knew the names, I knew something about the organisms, but so there was some exposure and awareness on my part.
As you became more interested in genetics and biology, were there any heroic figures in your mind in the field that you modeled yourself after?
The role model for me, scientifically, I think was my thesis advisor, Ethan Signer. Ethan was a young, very aggressive, very smart, very excited and into it molecular geneticist who was working on lambda integration and recombination. Let me tell you how I wound up going to his lab. So I went to MIT as a graduate student, and I went to MIT at the suggestion of or applied to MIT at the suggestion of Bob Edgar. And I applied all over to a variety of schools, and got into Rockefeller and MIT, and chose to go to MIT, and it was the smartest thing I ever did. Even though Bob Edgar knew that I wasn't that high on Caltech was difficult for me, but he still recommended I go to MIT, and he was exactly right. So I went there, and I went there in biochemistry, and the very first day I changed out of biochemistry into microbiology because it was very clear that's what I wanted. And then at the end of the first year, I remember standing outside of Ethan's door. I was intrigued by the opportunity to learn how messenger RNAs got activated, and Ethan said, well, with Lambda, you learn how to think. And that's something I understood immediately, and I really believe that. And I decided to work in his lab. And coming into his lab was really a flowering. He had a refrigerator full of hundreds of phage strains and a bacterial collection full of hundreds, if not thousands, of bacterial strains. Because Ethan, unlike the people at Caltech they had 3 bacterial strains and a lot of phage. They didn't do bacterial genetics at Caltech. But Ethan had been a postdoc with Jacob and with Brenner. And so he knew how to manipulate bacteria. And then, of course, Lambda obviously lives part of its life inside bacteria. And he came to, well, I tried a whole variety of things, but he presented me with a puzzle. He said, Ira, we have a deletion here, a prophage deletion strain, where the R gene is present. But when you infect, you cannot turn on the R gene. You could supply the Q protein to this prophage, and it had a deletion that ended in the S gene. The R gene must have been there, but it couldn't be turned on. He said, figure out what's going on. I mean, so here, this is in vivo biochemistry, working with activator proteins, Q protein, working with, the word promoter had just been coined by Beckwith. And here I was, being able to identify essential sites for turning on transcription, the promoter. So here are all the late genes here. The R gene is present, but it couldn't be turned on. But in the deletion here, which had a shorter deletion, you could turn on the R gene. So the first experiment I did, which really struck pay dirt, was R is regulated by this interval here. What about the other late genes here? So I looked at induction of the other late genes, in particular F and K, A, D, L, V, B, C, D, E, F, Z, U, V, etc., yep. F and K, and lo and behold, the same deletion interval which regulated expression of the R gene regulated F and K. And there it was, very simply in one fell swoop, that it looked like one regulatory site, one promoter, regulated these 3 representative late genes and probably all the late genes. So it was just phenomenal. I mean, everything was sitting in the refrigerator or in the backyard collection. If I thought of an idea, I just went and did it. And Fred Ausubel was a graduate student a year ahead of me, and he was working on int-promoted recombination. But like Ethan, he loved thinking about gene regulation, and we brainstormed endlessly about experiments to do.
So Bob Edgar was an obvious hero of yours. Who else played that role in your early career?
Well, in addition to the great graduate students that were in Ethan's lab, and that was Fred Ausubel and Martha Howe, were both ahead of me, and some other people. It turns out that Jim Watson was then at Harvard. He was very excited about Lambda. And so he also had just taken the position as being director of Cold Spring Harbor Lab. So the summer of my first year, so in 1968, Jim arranged for people in his lab and Messelson's lab, his lab and Potashny's lab, and to join Ethan's lab. And I spent my first summer in graduate school at Cold Spring Harbor. So I interacted a lot with Nancy Hopkins, who was Mark Potashny's first graduate student, and Jan Perro, who was one of Jim's graduate students. And those people were really up on Lambda and very steeped in biochemistry and wonderful people. And let me tell you about another one, Harvey Eisen, who was a folk hero of Lambda. And I was very fortunate to meet him. And anytime anybody had an idea, the retort was always, Jan Perro would have an idea, Nancy Hopkins or whatever. It would always be the same, Harvey has done it. So Harvey Eisen had done his graduate work at Toronto with Siminovich and First, and then was a postdoc in Jacob's lab. And Harvey's done it. And so amazingly, Harvey Eisen came to Cold Spring Harbor, and he spent a month at Cold Spring Harbor. I heard so much about this guy, and he worked in the same room as me. And he would grow up a culture, and here as a first-year graduate student, he'd take a culture and he'd hold it up and say, Ira, what do you think the OD is? Or how many cells do you think there are there? And every experiment he did was a spot test. And he had an incredible mind, and so he would interpret this in the most incredible ways. I think I have a song lyric which says something like interpreting, how is this going? You can't derive a dinosaur from just a single bone. Harvey could take, any spot test and make some fanciful model. And so basically it showed me that even the smartest person, they didn't always do the experiment to the nth degree. I mean, he sort of took it to a certain degree and had ideas. One should not be intimidated by the fact that Harvey was reputed to have already done it. And this proved to be a lesson I carried over very much into yeast. Let me come back and describe the thinking about my first year in Ethan's lab. He was interested in recombination and isolated recombinationless mutants of lambda and isolated non-integrating mutants of lambda, very super stuff. But I was interested in gene regulation, very much tied to why are early genes early, why are late genes late. And I thought of, a mutant hunt. The mutant hunt I thought of was lambda mutants that can make late proteins early. I understood the concept of genetics. So, you get something that, if it can't do it, you get something that can. So the idea was to take a late gene TS mutant, like a KTS mutant, and then if you do shift experiments to high temperature, there's a certain point that if you shift light enough, then you get a full load of phage. If you shift a little earlier, then you don't get phage, but anything can make the K protein earlier, blah, blah, blah. So what it did is it got my foot in the door on stuff that I really loved because it got me thinking about the late genes. That was really fantastic that I was already getting involved in doing experiments to test my little ideas.
Many of us have marveled at the effectiveness of the way you interact with graduate students. Tell us a little bit more about how Ethan interacted with you when you were a student.
Well, Ethan was very popular with graduate students, and he had a million ideas. And he started out by giving me just telling me, writing out the protocol of some simple experiments. And he was very informal, and he was just bristling with energy and ideas. I mean, the style of genetics, Jasper, that you know and that we do is one where you have hypotheses, and you isolate mutants, a lot of logic is involved, and we're comfortable with the biochemistry and with the genetics. And Ethan was very much like that. Lambda was fantastic because even though very few proteins had been identified, we were thinking in terms of the proteins. And as a genetic organism, Lambda was phenomenal. It was just like yeast is now. Lambda was the test bed for molecular genetics and molecular biology because you could make deletions of lambda. Mm-hmm. You didn't do it in vitro, you just did it. Lambda could be carried as a prophage. You could delete it. You could do all kinds of things. So it was very powerful in that way. We could do deletion mapping to find promoters and things like that. The reasons why lambda is so powerful. And then there was some biochemistry. Potashny did the important work of showing that the lambda repressor binds directly to DNA, and that's a real molecular touchstone.
Well, let's continue that line of thought for a minute. What were the other really exciting developments at the time that you were doing your graduate work that made a big impression on you and your thinking about your future career?
Some of the highlights of my graduate career. Well, this happened very early in my graduate career, but I remember Jacques Monod came and gave a talk at Harvard. In those days, the graduate students at MIT thought nothing of going over to Harvard or to Harvard Medical School and braved the rush hour traffic to hear speakers. Mm-hmm. We got to hear a lot of good speakers, and that was inspiring hearing Monod. who was describing about the genetics leading up to the lac repressor, and of course then the biochemistry emanating from the Harvard labs. Other things that, well, one of the greatest stories in Lambda, it was a source of endless fascination, was what Eisen found. At this time he was working in Jacob's lab, was that you could take a Lambda lysogen with a variety of mutations in it that carried a temperature-sensitive C1 mutation, so that at low temperature it was repressed and at high temperature it was not repressed. And you could, depending on how you cultured this strain, it could exist in two stable states. In one state, the C1 protein was functional and the cells were immune to superinfection. And in the other state, the cells were not immune, as if the C1 protein was not made. But the same DNA was there, but it's stably immune or non-immune. Now that was interesting. There was a second repressor, CRO, which inhibited synthesis of C1. C1 inhibited CRO, CRO inhibited C1. So I knew that these kind of stable states and molecular switches were interesting. This was the height of what's interesting. And I should say that one person I interacted with a lot when I was at MIT was David Botstein. And David came first as an instructor, and then he was soon appointed as an assistant professor. And David was working on many, many different aspects of P22. And some of his best work, I think, in all of science was done in that period. Not that he hasn't done terrific things since then. But he did some amazingly beautiful work on gene regulation in P22 that nobody knows about, because it sort of, just as his work was reaching fruition, people were moving on from Lambda to eukaryotes. But his studies on the mechanism of repression control with an anti-repressor and then a repressor that regulated the anti-repressor, those puzzles were just phenomenal.
I wanted to ask you about your first presentation of your own research at a major meeting.
I remember my first presentation of scientific work was at a group meeting, at the Edgar Group. And I was describing the work on these cofactor requiring mutants. And I said a few things like, well, these are the results I got, but I don't really believe them because the numbers were small or whatever. And afterwards, the graduate students there took me aside and they said, Ira, you just present the data. We'll decide whether we'll believe them or not. The first talk I gave as a graduate student, I'd done this work in identifying a late promoter or an essential site for late genes in Lambda. And I was invited by the Watson Gilbert Patashny Messelson group to give a seminar there at the biolabs. And I never forget, I don't remember the talk, but I remember the night before having this panic attack that maybe what I was proposing wasn't true. So I did the experiment again the night before the talk, and the talk came off fine.
So you had a fairly unusual, by modern standards, route from the end of your graduate work and a short postdoc to your job in Eugene. Tell us about that.
I entered graduate school at MIT in 1967, and things went very well. And I went to the Cold Spring Harbor Phage meetings, and I guess gave several talks, and people knew who I was. And amazingly enough, I got a job offer from Frank Stahl in my 4th year of graduate school. So typically the molecular biology, molecular genetics hotshots would go do a postdoc in Europe at the Institut Pasteur or the MRC or in Geneva. And that would have been the sort of reflexive thing for me to do. But I didn't have any desire to do that. I didn't know what I wanted to do exactly. But I didn't particularly want to go there and try to figure out what to do for 2 or 3 years. And I saw a paper, 2 papers that influenced me. One was a paper which claimed to find viruses in a mold. And I thought, aha, a simple eukaryote with virus, that would be good. And then I saw a paper from Tom Manny's lab, which was yeast alpha cells make alpha factor, and they make halos on a lawn of A cells. So I can really relate to plates and holes in plates, since I spent my whole graduate career doing that. And so I said, okay, then when David said something about the Cold Spring Harbor yeast course, we took it together. So I took it in the summer of 1971, and I got my PhD in September of 1971. So I took it before I got my degree. Some of the things I remember from the yeast course at Cold Spring Harbor, first of all, the contributions and the effort made by Jerry Fink and Fred Sherman cannot be overstated. I mean, I guess I took the yeast course in the second year, and it was phenomenal. They had all kinds of people passing through, and one person who passed through was Tom Manny. So Tom Manny talked about mating type, and particularly talked about alpha factor and the fact that he and his graduate student Vivian McKay had isolated mutants that didn't mate, and they showed that some of them were defective or A-specific and alpha-specific or non-specific. And he said he didn't have anything written up about it, so he said, Ira, I'll give you my grant proposal. You can just, it's all written down there.
Wow.
And of course he sent us, well, the strains in our strain collection, Vivian McKay sent them all to us. So his generosity was phenomenal. So I came back to MIT, and then the first semester I worked on yeast. And the first thing I did was the reason I chose yeast was twofold. One, to understand why alpha cells make alpha factor. A cells have the gene, but they don't, I presume they had the gene, I didn't really know, but why do alpha cells do it, A cells don't, why is that? That should be something that one can figure out. Second thing was, I discovered Hawthorne's 1963 one-paragraph paper on switching from alpha to A, and all by deletion, and that made no sense. It's obvious that alpha and A are codominant, mat A and mat alpha are codominant. So how can you get a deletion that makes that switches an alpha to an A? I didn't understand it at all, and I thought, well, a mat deletion is it's a null mutation, and so it should be possible if you can delete mat alpha and make it into an A, you should be able to make a nonsense mutation. It could do the same thing. So that was the first thing I tried to do in David's lab, is to switch an alpha to an A by a nonsense mutation. Of course, it didn't. But it got me exposed to what happens when you do rare matings. After 1 year at MIT, where I was in David's lab, but I was an instructor, then I went to Eugene. Another person who was very, very encouraging for me getting involved in yeast was Lee Hartwell. And I actually had a couple of interactions with Lee. When I was a senior at Caltech, trying to decide where to go to graduate school. And then many years later, when Lee heard that I was coming to take a position in Eugene, he said to me, he said, anytime before your lab gets set up that you might want to spend in my lab, you're welcome to do so. And then he invited me to a meeting that they held that summer. I guess it was the summer of 1972.
So when you started your lab in Eugene, you didn't start with yeast, which is sort of odd considering your obvious excitement about the yeast work you were doing as a postdoc. Tell us about your thought processes and which projects you started your lab with.
Well, when I got to Eugene, people had been preparing for my arrival. Frank and Mary Stahl had cleaned out my lab themselves and made it perfect move-in condition. And then Carol Gross, who had been a graduate student and a postdoc with Pete von Hippel, had been an instructor in a molecular genetics course. And she had primed the graduate students there for my arrival. And she had told the appropriate students to go work with me. And then she told me who I should take.
Wow.
Yeah.
So it's a fixed job.
At any rate, I instantly acquired 3 graduate students, Jim Hicks, Jeff Strathern, and Penny Toothman. And basically, the lab was not set up, and so I said, Jim, here's the microscope, here's a micromanipulator, why don't you get them set up? I had no clue how to do it. And then after he did that, I said, we have this strain from the Espositos, that has this D allele, this diploidization allele. Let's just see how to score it. I don't understand it. It's somehow involved in homothallism, whatever that means. Do a cross and see if you can score it in the cross. So that was the start of everything. He did a cross to a strain which was didn't diploidize and was able to follow the marker by doing standard plate assays and got him to think about it. So Jim was going on yeast. Meanwhile, Jasper Rine, very, very smart guy, was doing very, very rarefied experiments with lambda, because you could manipulate lambda and all kinds of clever things. And he was doing a variety of things. I forget exactly what they were, but they weren't very interesting and they were going nowhere. He wanted to be working on yeast, dissecting cells methodically, doing the pedigrees, and then testing their mating type by seeing whether they responded to alpha factor or not. It was Jeff's suggestion to look to see the response. If you interpose A cells as a barrier, does that interfere with the test? Real simple-minded stuff. The original goal was to understand, as I said, the mechanisms of gene regulation, which has always been a theme of interest of mine. So why do alpha cells produce alpha factor? And that was a big hand to play out. And the second one was mating type switching, because, gosh, any switch has got to be interesting, right? So, I mean, those are two big things to play out. What made all that work take off and us to understand the mating type locus was the fact that Jasper Rine was able to take fragmentary information from over here and fragmentary information over here and put them together. And in this case, the fragmentary information was there are all these weird mutations that were at the mating type locus, and then this weird stuff from Anita Hopper and Yona Kassir about mutations that bypassed, that allowed AA cells and alpha-alpha cells to sporulate. So Jeff took those mutations which bypassed the mating type locus and allowed things to sporulate, and he applied them to the mutations of the mating type locus. Oh my goodness. He was using, you know, in the hands of a smart person, he used one unknown to study another, which usually turns out to dreck. For Jeff, it turned into gold, because then he was able to cross an alpha-1 mutation with an alpha-2 mutation and predict, amazingly, that the double mutant would mate like an A and blah, blah, blah. There's nothing, that's got to be the high point of, you know, of scientific logic, or genetic logic, certainly in my lab. That was awesome. And then another great moment, you'll know the details better than I will, since you were responsible for it, but you came into the lab at a time when Jim Hicks had found some kind of suppressor mutation which allowed alpha-1 mutants to mate. And then you showed it suppressed an alpha-2 mutation. Now that was confusing. And then you were on a roll, so then you showed it suppressed an A1 mutation. That was totally confusing. But it completely said, we don't know what's going on. There's something here to figure out, and I had absolute confidence it would be figured out. I mean, that's what I meant when being in Eugene was like an Alpha in Wonderland, an Alice in Wonderland moment. All these things were going, you know, Alice in Wonderland, and you look up in a tree and you see this weird thing in a tree, right? And it's amazing. And then the chronology was, I mean, then at some point the hypothesis came about that these genes required for mating, for homothallism, might be silent versions of the mating type locus. And then you rapidly did the obvious genetic experiment and showed that it was all true, and then everything made sense. And so, I mean, it was a great group. I mean, you and Jim and Jeff, it's hard to imagine a better group than that working on one problem. I don't ever remember any friction. And if anything, it was just handing things off. Yeah.
Did Oshima's work influence your thinking at all about mating type switching?
Well, of course, Oshima's work was the coin of the realm. I mean, it influenced us in a couple of ways. He did this beautiful work of showing that to change from mating type from A to alpha required HO, and to go from A to alpha required something that he called HMA, and then go from alpha to A required HO and HM alpha. And then you know about HMA and HM alpha because there are defective versions of the strains. And then the result that was just too confusing for words. The double mutant, defective in this one and this one, could switch in both directions. Then Jeff Strathern, of course, proposed that the HMs were not cofactors, they were the information, and it was moved over. So, Oshima's work provided all the stuff we thought about. Yeah, I mean, it was great, and he did pioneering great work. It's just that it didn't make the links that we made. We moved it ahead. I wanted to make a couple of more comments about how working in Lambda prepared me in a natural way for what was coming in yeast. And by analogy, I think people who are trained in yeast are prepared intellectually for other challenges. But in the case of Lambda, a couple of things. I remember when I was a graduate student, the phenomenon of prophage curing. You have a Lambda prophage, it's sitting there, a block of DNA. And if you superinfect with a phage, a Lambda immunity 434, that's the same as Lambda but doesn't, has a different repression, it can replicate in whatever. And lo and behold, among the survivors of that infection are cases where the infecting phage has replaced the prophage. It's a prophage switch. It's exactly like mating type interconversion. And the other thing, very importantly, in Lambda, we always had the attitude that we can figure it out. You ask a question, you can figure it out, no matter how bizarre it might seem, how complicated, how this and that. You just do it.
I remember a couple other projects that you were interested in at the time. In fact, one led you to grow ladybug beetles in the lab.
Yeah.
Tell us what you were thinking about in those projects.
Well, interesting that you remember that. Well, ladybird beetles, I was at Cold Spring Harbor sitting on the grass outside Barbara McClintock's office, and she came by, and Barbara had made connections with me. I'd made connections with her long earlier because my father, when he was at Missouri, studied with L.J. Stadler, but also he took classes from Barbara.
Wow.
And he said that she was a great teacher and would take the papers and underline all the important parts of the papers for the students. She was a very good instructor. At any rate, so I always looked her up when I went to Cold Spring Harbor, and she loved me because I was a geneticist. And so she said, well, what are you doing? And I said, well, we work on, you've heard of Jasper, we work on yeast mating, and there are 3 cell types. There's A's and alphas and A alphas, and it's determined by the mating type locus, and the mating type locus, the alleles are codominant. That's the last word I got to say was codominant. She took over. So then she riffed on codominant alleles, and one of them is pattern formation in ladybird beetles. So you have the, on the wings here, these 2 spots here, and then you can cross that with one that has 1 spot, and the hybrid then has a superposition. So it's some kind of pattern formation determining locus, and work by C.C. Tan, this great Chinese geneticist. So that made me interested in maybe, if you can do Drosophila genetics, you should be able to do ladybird beetles. And so that was the genesis of that.
Did you ever get the beetles to cross?
No.
One of the things that has dazzled me over the years, well, there's 2 things actually. One is the judgment that you exercise in terms of picking new projects, and the other is the way you pick people to work in your lab. So let's talk about those.
In Eugene, it was real clear what those projects were, mating type and mating type switching, and they grew a life of their own. And I'm not going to go through all the different steps in slow motion, but the people in the lab, Rob Jensen and George Sprague, adopted cloning methods and were able to get their hands on the relevant genes, which then made biochemistry possible. And then Sandy Johnson, who had trained in Patashni's lab. I mean, can you imagine when you want to go make the jump to biochemistry to have one of the great graduate students from Patashni's lab come to your own lab? So that was, doesn't get better than that. But then after I moved to Eugene, left Eugene and went to UCSF, this is cell biology city, and so the gene regulation things were being handled very well. But in this milieu, then graduate students like Fred Cheng and John Chant just went at it in terms of getting involved in cell biology, where Fred was an MD PhD student interested in something relevant to cancer, and so he started to work on this idea of how do cells respond or get arrested in response to alpha factor, and his work is stellar. Fred did a whole year rotation, worked a lot in my lab in his first year of medical school, played a lot of hooky. And then, of course, my whole yeast career has been the 3 different cell types and how they differ from each other, and one of the differences is in the budding pattern. Haploids bud axially and A-alpha diploids bud in a bipolar manner, and I remember telling John Chant, you should isolate mutants that don't bud in the right place, and he struck big pay dirt. I mean, John's mutant hunt could not have been more revealing.
Let's back up a second and talk about the move from Eugene to UCSF. You were obviously one of the key people in the department there, very successful, well-liked by everyone. That must have been a difficult move for you.
Well, I obviously thrived in Eugene. I was there for 9 years. Scientifically, I could not imagine a better group of people, and the graduate students were the engine. And it turns out that the top quality graduate students stopped coming to Eugene. They had come to Eugene because they were people trained in sort of quantitative or chemical sciences. In your case, you were neuroscience, but you're used to rigorous thinking, who wanted to do something biological. And the original Institute of Molecular Biology had Stahl, Streisinger, and Novick. I mean, you can't find a better group of molecular geneticists than that. And then I spent a sabbatical here, associated with Keith Yamamoto's lab, and here there were a lot of people doing eukaryotic cell and molecular biology. In Eugene, there were none. So the graduate students weren't coming in Eugene. There was no cell and eukaryotic cell and molecular biology, and I was on a collision course to be department chair. And then I had personal reasons because to move out of the area. So all those things combined, it was natural, and they were very interested in genetics here. So I think it was the right thing to do.
Looking back, we've already discussed several of the things that you were quite excited about, but of the many successes you and your lab have had, which ones have given you the most personal satisfaction?
There's some things that stand out because they're very distinctive. Like Jasper Rine's paper on the switching pattern of homophthalic yeast. This is a paper published in Cell, which had 1 strain, nothing was cloned, no mutants. And so the point is that there was a very, very rich intellectual, very intellectually rich paper, just pure description, and it was fascinating. There were various phenomenal spin-offs like repression of the silent cassettes by Sir, which has turned out to be a whole important aspect of gene regulation itself, and then all the way to why mother cells switch and daughter cells don't, and identification of ASH1 and RNA localization. So it's wonderful to have a story sustained over the years like that. And then the mating type locus, I mean, the mating regulation of cell type by the mating type locus. Now it's also cut and dried and clear and this and that. I mean, this is the way you would, this turns on this and this turns off this, and it's all very straightforward, and we've identified it, and all the biochemistry's been done by Sandy Johnston, and I mean, that's a great story, and it's textbook stuff, so what's not to like? And then John Chant, who went after a problem of budding pattern, again, by the most brute force, mundane genetics. Let's just get mutants that don't bud right. And he did a phenomenal study, which nothing cloned. He just had mutants and double mutants, and he proposed a whole pattern, the whole pathway by which budding pattern works. Landmarks and organizing the cytoskeleton. I mean, all the ideas were there in one beautiful genetics paper. I was head of the genetics program here for 20 years, and one of the greatest satisfactions about being at UCSF is that we don't feel we have to flag way for yeast genetics here, or Drosophila genetics, or nematode genetics, because it's abundantly clear to all of our colleagues here what genetics is good for, and they've put it to good use. For the last 4 or 5 years, I've been the co-director of the program in human genetics here, my co-director being a true medical geneticist and mouse developmental biologist, Charlie Epstein. And so what we've done in this period of time is to try to to build up the infrastructure and the community of researchers here in human genetics so that human genetics will be in the fabric of the whole community here, the clinical community as well as basic science. And my own personal involvement with human genetics is in a particular aspect of human genetics, which is pharmacogenetics. And a colleague of mine, Kathy Giacomini, who's the head of the Department of Biopharmaceutical Sciences, she wound up writing, being PI of a massive grant in pharmacogenetics that I'm co-PI on. And so now we're really heavily involved in looking at transporters, integral membrane transporters, as candidate drug response genes. And it's gotten me to thinking about some things I'd never thought for a moment about, namely population genetics. So this is very different from microbial genetics, but I'm learning. I mean, I am a geneticist. I want to be involved in human genetics, and this is the way I'm doing it.
You're also obviously well known as being one of the most gifted teachers of genetics in any field. What role has teaching had in your research career?
Well, I've always considered the profession being as a teacher. I'm not here just as a scientist to work in the lab and churn out as many papers as I can and make myself famous, but as a professor and as a teacher, I think that teaching is the highest level of intellectual activity, more than discovery in the lab. Isn't we able to tailor the story to reach the people? And I think that's a tremendous challenge. That's the greatest challenge. And it's what, like Bob Seger says, you have to know what to leave in and what to leave out. That is really, so my experiences as an undergraduate have taught me a few things about teaching. One is that too often the educational system has people, rubs people's faces in what they don't know. People are made to feel inadequate because they don't know something. So I think it's important for people to feel good about what they do know and not to feel overwhelmed by the certain things that they don't know. So in my teaching, I always am careful to start at the beginning and give people a chance to get on board. And I don't try to flaunt any brilliance, just try to be clear. And I'm a great believer in using graphics to complement the words. And you know, certain things that people violate all the time, like don't use terms unless you've defined them. So these are simple things, and people feel good when they learn something. It's like when you're singing a song and the chorus comes around again. Doesn't it feel great? Here it is again, and the circle, they go round and round, and the painted, you know, or you can get anything you want at Alice's restaurant. I mean, as it finally comes around again, everybody's very happy. Yeah. And like you give a lecture, and you know this because you teach undergraduates, that you give them a tool, and then you come back and ask a question later, and then they all say, oh, I use that tool I just heard about. It's a tremendous feeling of satisfaction and feeling that they understand something. And it will make some sense to you in light of everything else I've said. There are two scientific high points of my career. One was Francois Jacob asked me to give a set of lectures at the Collège de France. And so I lectured on yeast mating type, and he was in the audience, and my style, as you know, is to ask questions of the audience, and he was sitting there, you know, raising his hand. So to have one of my scientific heroes actually to invite me to give his set of lectures was a great thrill. And the second one was a few years ago, about 5 or 6 years ago, before David Baltimore went to Caltech. The Caltech biology faculty decided they needed to educate the physical scientists on campus about biology, which they considered a soft science, you know, the physicists, etc. So they organized a series of lectures in biology called Biology 101. And I was the only outsider invited to give a talk. So I gave a chalk talk on lambda. And sitting in the audience was like my physical chemistry professor, Aaron Cooperman. It was quite memorable. That was quite, really closed the loop, yeah.
What advice would you have for somebody that's a graduate student at this point thinking about a career in genetics?
Oh, I would be absolutely 100% supportive, but I'd also make it clear that being a scientist is like being an artist. You have to want it. It's not a downhill slide. It's a lot of challenges and uncertainty, but you have you're your own boss, and you get to be around, live a life of the mind and be involved in teaching and learning. Oftentimes I've gone to give a talk somewhere and you meet somebody and you say, what do you do? They say, I'm just a graduate student, right? And then I say, well, after you're just a graduate student, what? You're just a postdoc? And after just a postdoc, you're what? Just an assistant professor, etc. And then I go into my little shtick here, which is something that I learned from my English teacher in high school, Mrs. Gottlieb. And in the back of her classroom, on the far right, was a little inscription that says, this is not a dress rehearsal, this is it. The fact that our students can really be doing science, I think is so wonderful. And then people sometimes need to be reminded of that. Mm-hmm. And so they say, I'm just a graduate student. I won't stand for that.
What's changed the most in your perspective of genetics and a career in science between the time that you started and now?
Oh, I think about that all the time, and I feel sort of a little intimidated by science right now, which is to say, when I started, keeping track of lambda or keeping track of yeast mating type was trivial. I knew everything about lambda. I knew everything about yeast mating type. And that was a great feeling of command. And now it's just a joke. There are things going on in my own lab I have not that great a grip on. And trying to, the fact that I'm interested in cell morphogenesis and cell cycle regulation and gene regulation and just within yeast and sporulation, it's overwhelming. And, you know, on a daily basis, I get 1 million journals, and gosh, I barely have time to put them on the shelf. How do we keep how do we keep some kind of a perspective when there's so much stuff out there? So that's one thing, and there's so much stuff out there because it's easy to do experiments now, and this is the other thing that's intimidating about current science is the whole genome analysis means that for a lot of people there's a whole technological barrier until they can get kind of certain kind of experiments done. And the experiments that for me have been the hallmark of my career have been the simple things on plate assays and agar block experiments and, you know, of course, northerns and protein purification and southerns and this kind of stuff and looking under the microscope. But I'm a little worried about, I think the cottage industry aspect of American science has been very important, very successful, and I hope that the technology doesn't get to be a barrier. I consider myself very fortunate to have, one, grown up in the tradition of microbial genetics. I really am cut from the cloth of T4 and lambda, and I don't think one can do, I'm very proud of that, as you can tell.
Oh yeah. Well, Ira, this has been really great fun for me, and I'm sure it's going to be enormously interesting and informative and educational for all kinds of people. It's really great that you were willing to do this. Thanks.
Oh, well, I mean, it was a blast, thrilled to be honored. I wouldn't have wanted anybody other than you to do it. I began playing the guitar as a sophomore, and it was strictly a sanity preservation device. My twin brother had started playing in his freshman year, and I just didn't have any interest in doing it. But then I saw Bob Dylan perform in the summer after my freshman year, and I was very inspired. Come gather round, people, wherever you roam, and that, you know, that stuff. And so I got some cheap guitar, and Joel showed me a few chords, and I sort of learned a whole bunch of Bob Dylan songs and things like that, and entertained a little bit at various campus functions, and also became somewhat of a folk music entrepreneur because they had a series at 11 o'clock on Wednesdays where folk musicians could come. So I brought in Jose Feliciano and Buffy St. Marie and Sonny Terry. So I got to meet some of the famous people there. I've never given a concert at Caltech. It'd be something I would like to do. Although I was at a dinner partially in my honor there, and during the dinner, attended by the president of Caltech and everybody else, I was able to borrow a guitar from some undergraduate in the dorm, and I sang the Caltech Talking Blues right there for the whole masses. Well, when I was a kid, I had this dream to be on Caltech's basketball team. I dropped by the gym to jump and shoot, but no one around seemed to give a damn. All too busy, I guess, writing folk songs. Well, my 4-year ordeal had just about passed, so I looked for a grad school where I could have a real blast. I applied all over, and you know what to expect. I got turned down everywhere, except Caltech. In Eugene, I had a renaissance and interest in playing guitar, and there was a Willamette Valley Folk Festival. And I really, really practiced up for that. And I remember the morning of the concert, I went over to the gym where it was indoor auditorium, and I was so nervous, my knees were weak. The last time I felt like that was before a track meet in high school, where I was a 9th rate 440 runner, quarter miler. But oh, was I nervous! Oh my! And during that performance, I had a glass of water, and in typical folk tradition, they filled it partly with whiskey. What I didn't realize, as I kept on drinking it, and it could always stay level no matter how much I drank, they kept on refilling it with whiskey. So when I left the stage, I could barely walk, and I was so drunk, I was drunk the next day. At any rate, it came off well. I was very happy. I did it again the next year, and I practiced, and it came off well. And I did it the next year, and I practiced. It came off pretty well. But the high was the biggest high was the first year, because the thrill of doing a good job was directly related to how much anxiety there was in it. And the people in Eugene, especially Mary Stahl, just were always urging me to do it. And Mary died an untimely, premature death a couple of years ago. I would like to have done that again, and if I had done it again, it would have been for Mary. Yeah. Then I started to write songs. At UCSF, every year there's a graduate program retreat where the students put on a skit. And the skits are hilarious. They roast various faculty. They explain to the students what's going to happen to them. Terrifies them, of course. And the faculty is always threatening to do a skit. So I've got it together and wrote a whole bunch of songs that bear on different aspects of the life as a professor or what students are going to face. Like Shop Around is a song about how students better, check out the different labs before they commit to one. So that's one song. Another one is about, procrastinating writing papers, and another one is a grant proposal blues. So we sang some of those songs, and I've had occasion to sing more of those songs at meetings and things like that. It's fun. Yeah. Music is a wonderful way to reach people. Now, Judge, Your Honor, hear my plea before you open up your court. Now I don't want no sympathy because I cut my good man's throat. I wrote these songs about 10 years ago, and I called it the Fall Collection. And I wrote a whole bunch of songs about the experience of being a faculty member. And also, I'll sing one called Shop Around, about doing lab rotations. So, and the general idea of all the songs, basically, I grew up with rock and roll and rockabilly. And these songs capture the feeling. So this one, if I can do it successfully, you'll get the same idea. It's called Grand Proposal Blues. I'm gonna raise a fuss and I feel like I could holler. My lab is gonna close lest I raise another dollar. Sent off my application, went out to celebrate, but they said, no dice, son, we don't accept proposals late. Now I'm just wondering what I'm gonna do, cause there ain't no cure for that grand proposal blues. My accountant's always telling me you gotta get more money. If you want to do experiments beyond the next Sunday. Well, we tried to buy some enzymes. The vendor said to me, we'd like to help you out, but you know they ain't for free. I'm just wondering what I'm gonna do, cause there ain't no cure for the grand proposal blues. Gonna take 2 weeks, write a killer application. Gonna solve all my problems, get my final vindication. I sent in my proposal and the study section said, we'd like to help you, Ira, but that research area is dead. Now I'm just wondering what I'm gonna do, cause there ain't no cure for that grand proposal blues. Now here's one I rarely sing, because I may screw up a little bit, but it's a funny, funny song. I wrote this, it's called After the Banquet, and it's what it's like to be a speaker in the session that takes place after the banquet. And I actually had a chance to sing this at Cold Spring Harbor, because I gave a concert after the banquet. So yeah. Oh, we've been here for a week, 100 talks were more than enough. Now our backsides are numb and our tired minds are overstuffed. After the banquet, my body's weak, oh yeah. But I'll be getting up early, that's when I speak. It's the last session. It's at 8 a.m., the last session. There'll be an audience of 10, the last session. I got bad luck again, the last session. At the very end, the last session. Last one, the organizers claim they saved the best stuff for the end. You can almost hear them laughing as they use this line again. After the banquet, everyone's asleep, oh yeah. In an empty room, that's where I'll speak. The last session, that's where I'll be. The last session, everyone's asleep. The last session, you can find a seat. Last session, it's just the chairman and me. The last session, last one. Never heard that one, huh? So this is another one that was written especially for the graduate program retreat, and it's about laboratory rotations. And the head of our umbrella graduate program at the time was Mike Bishop. So that's why you have Mike Bishop playing a role in this. Near the end of my first year, Mike Bishop called me to his side. He said, bioreg is almost over. Pretty soon alive you will decide. And then he said, just because you've rotated in that lab now, there's still some things that you may not understand now. Before committing yourself to a lab now, keep your freedom just as long as you can now. Mike Bishop told me, you better shop around. Oh yeah, you better shop around. Before you choose a lab and say I do now, make sure that it's the one for you now, or else you'll wind up feeling sad and blue now. Mike Bishop told me, you better shop around. Oh yeah, you better shop around. You gotta get yourself the finest one. Don't be sold on the very first one. There's 3 or 4 labs that everyone is yearning, and 2 dozen more where you can get a lot of learning. You got to visit, talk with people in the lab now. Find out if they're excited about the research project now. Are their experiments always on their lips? Or are they only talking about the recent camping trips? Mike Bishop told me, you better shop around. Oh yeah, you better shop around. Consider more than the usual stiffs. A herd mentality can lead you over cliffs. Check out the younger faculty, they're doing what they oughta. The older ones can barely keep their heads above the water. Before you choose a lab and say I do now, make sure that it's the one for you now, or else you wind up feeling sad and blue now. Mike Bishop told me, you better shop around. Oh yeah, you better shop around. You better shop around. And Mike Bishop actually sang that in that. He sang the microphone. He was horrible. He can't sing at all. Yeah. Well, this is pretty self-explanatory. It's called I've Been Working on the Genome, and there's a chorus, which I'd like to hear some accompaniment on, which is a megabase to go, a megabase to go, a megabase to go till dawn. Okay? I've been working on the genome all the livelong day. I've been working on the genome just to pass my time away. Can't you hear the buffers flowing, gels from dusk till dawn? Then you read the sequence ladders, it goes on and on. A megabase to go, a megabase to go, a megabase to go till dawn. Megabase to go, a megabase to go, a megabase to go till dawn. Someone got the Huntington gene, don't they? Someone's got the gene, I know. Someone's got the Huntington gene, don't they? Now they're calling up the New York Times. Oh, I've been working on the genome all the livelong day. I've been working on the genome just to pass my time away. Can't you hear the buffers flowing? Gels run dusk till dawn. Now you read the sequence ladders, it goes on and on. A megabase to go, a megabase to go, a megabase to go till dawn. Megabase to go, a megabase to go, a megabase to go till dawn. We're walking down chromosome quattro. We're walking, but it's mighty slow. We're walking down chromosome quattro. Sequencing as we go. We're reading A, G, C, C, T, A, 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 A. Sequencing junk DNA. Oh, I've been working on the genome all the livelong day. I've been working on the genome just to pass my time away. Can't you hear the buffers flowing? Gels run dusk till dawn. Then you read the sequence ladders. It goes on and on. This is one of the first people, when I was in high school, a friend of mine, her parents had a Lead Belly album. And Lead Belly really impressed me. He played a 12-string guitar. and I could do Lead Belly imitation, which I probably will. Anyway, it's take this hammer, wah, carry to the captain, right? Take this hammer, wah, carry to the captain, wah. Anyway, that's the Lead Belly. And now we do take this toothpick. Ready? Take this toothpick, wah, test more colonies, wah. Take this toothpick, wah, test more colonies, wah. Take this toothpick, wah, test more colonies. I dream about mountains 1 millimeter high. If she asks you, wah, got any mutants, wah. If she asks you, wah, have you got any mutants, wah. If she asks you, wah, have you got any mutants? Tell her no mutants, everything is just a wild. If she asks you, wah, was the jail running, wah. If she asks you, wah, was the jail running, wah. If she asks you, wah, was the jail running? You tell her it was flying. You can tell her it was flying. I don't want no, wah, DNA sequencing, wah. I don't want no, wah, DNA sequencing, wah. I don't want no, wah, DNA sequencing. It dulls the mind, it dulls the mind. No, I wrote it. Oh, I know, I don't even know, need to look at the lyrics, I know that. You ready? I got a fine laboratory, sharp people inside. They work day and night, they think far and wide. When they get an idea, they don't make no excuse. Grab hold of the problem, turn it every way but loose. I am a molecular biology man. You know my neighbors are confused, but my colleagues, they understand. You know my favorite hypothesis, shot full of holes. I told the people in the lab, I want to see those gels roll. Student in the lab, she's doing her PhD. Committee said, let that woman go free. I am a molecular biology man. You know my neighbors can't figure me out, but my colleagues, they understand. I got a policy. I went to a meeting, I learned a lot I didn't know. I heard the same talk 7 times in a row. I talked with a competition, we played our cards close to our chest. We talked about the weather, gonna publish all the rest. I am a molecular biology man. You know my neighbors are confused, but my colleagues, they understand. You know, I got an old-sized microscope, an ABI machine. Incubators at all temperatures, high and low and in between. I got a brand new project, don't know if it's gonna fly. But if it does, I'm gonna be sky high. I am a molecular biology man. You know my neighbors are confused, but my colleagues, they understand. Let's help me out now. Give me an M O L C C U L A R. Molecular biology, we do some enzymology, man. Molecular biology, DNA technology, man. Molecular biology, it's a branch of astrology, man. So I've written several songs really trying to capture what it's like to be a practicing molecular biologist, molecular geneticist. And one of the things that we do is write a lot of papers. And I wrote this song at a time when I had 3 or 4 paper commitments to Cold Spring Harbor Laboratory. And so this song is called I'm Ready to Work on the Paper. And I think it's a song about procrastination, so it transcends writing papers. I don't care how much time I gotta spend, got to find the surface of my desk again. Piles of mail are tossed, I found the things I lost. I'm ready to work on that paper. Oh well, the paper's overdue, they said they couldn't wait for me anymore. Listen, Joan, can't you see I'm gonna get back to my writing once more? Any day, don't know how much more pressure it's gonna take. Got to sit down and write for heaven's sake. The boom's about to fall, my back's against the wall. I got to work on that paper. When I agreed to write the paper, the due date seemed as if it's never gonna come. But time has passed and deadlines missed, always something else to do, but now it's #1. Every day, yeah, give me an hour on an aeroplane, or a minute at the elevator waiting for 9. Words are flying fast, the paper's done at last. I'm ready to work on the next one. You know that paper's overdue, they said they couldn't wait for me anymore. Listen, Joan, can't you see I'm gonna get back to my writing once more? Any day, yeah, I don't care how much time I gotta spend. Got to find the surface of my desk again. Piles of mail are tossed, I found the things I lost. I'm ready to work on the paper. You know that paper's overdue, they said they couldn't wait for me anymore. Listen, Joan, can't you see I'm gonna get back to my writing once more? Any day, this song doesn't have an end. So the editors that are mentioned in this are the old editors. So the old editors are Ben Lewin, Dan Koshland, John Maddox, and Jan Drake. Is this okay? I sent the paper to sell, I sent it special D. I had visions of a cover, but it came right back to me. Then wrote upon it, return to sender. I read with a groan, not enough data, nothing cloned. I tried to shorten the manuscript, add the data that it lacked. I tried to revise it, but my paper kept coming back. I sent the version to science, I sent it Federal Acts. I hope to feature it prominently, but I think this paper's heck. Dan wrote upon it, return to sender. Not much to discuss, please don't be offended. It's just not for us. We isolated a dozen more mutants, we cloned and sequenced 3 genes too. But the paper was sent right back, never went out for review. I sent the paper to Nature, I sent it DHL. I had visions of an article, but it came right back as well. John wrote upon it, return to sender. Not of interest to me, needs more color, that's COLOUR. Try JMB, sent the paper to I sent the paper to genetics. I sent it priority mail. I had visions of acceptance. I hoped it wouldn't fail. Jan wrote upon it, this paper's accepted. It's a lovely tale. Data are substantial. Proofs are in the mail. Our paper told a beautiful story, but it wasn't for the average Joe. Maybe someday the masses will discover what we already know. That this paper's a classic, I think you get my gist. If they open genetics, They're going to see what they missed. So this song was written by my twin brother, and I was a first-year graduate student at MIT, sitting in a lab that David Baltimore had just come into. And it was around, well, it was winter, 1968, and my brother was in his first year of medical school, and he had written 1 million songs in his first year of medical school. How that's possible, I don't know. But he did. And this song is nothing short of a masterpiece, the Double Talking Helix Blues. Well, I once saw a Charles Adams cartoon. There's a Martian sitting in a waiting room. It was late at night in the maternity ward. A nurse appeared as she opened the door. "Congratulations," she said, "it's a baby." Well, the point of the story I'll tell you right now. Did you ever sit down and think about how it is that every time a baby's born, it's a baby, not a rabbit or an ear of corn? Well, it just so happens that inside every one are tiny plans to tell how the job's to be done. They're worth more to you than the family jewels. They're stored in the form of molecules like everything else, I guess, only different and kind of special. Well, floating around in each one of your cells is a nucleus, and you know what else? Tucked inside every one of these are chromosomes that hold the key to the question we ask about how humans beget humans. Well, these thread-like chromosomes contain a groovy little substance that we're going to name. It's a macromolecule, as they say. It's mighty fine stuff. It's DNA. That's deoxyribonucleic acid for those of you out there with expanding minds. Now, this DNA consists of a chain of sugar and phosphate over and over again. But just a minute's thought and you'll see there's no hope for such a simple molecule to contain all the dope to get the human show on the road, that is, and into high gear. Well, each of the sugars in the backbone has got a ring-shaped base attached to it all its own. There's only 4 in a human being: adenine, guanine, cytosine, and thymine. They make up an alphabet of 4 letters: A, G, C, and T. I wouldn't want to write a novel with 4 letters. I think I'll write a human being instead. Well, there's one important fact we've let slip by, and it earned 3 men the Nobel Prize. It has to do with 3-dimensional conformation and how it relates to information. You see, DNA consists of 2 strands joined by the bases, holding hands in the form. Hydrogen bonds, that is. Any question about this? The whole molecule forms a double helix, a spiral staircase arrangement with the nucleotide bases represented by the steps. Hope this isn't too steep for you, Jasper. Now these bases pair in a special way, C with G and T with A. The reason for that's not too complex. I suggest you look it up in your text. You know the book. Some of you may even have opened it. Now here's a Nesselson-Stahl experiment. Now sometime before a cell divides, an unknown factor in the cell decides it's time to copy the DNA so the new cell will know the way, how to survive and do what it has to do, a lot like us. Hence the double helices unzip and nucleotides in the neighborhood slip into place next to their proper mates. I think you see what this creates. 2 daughter macromolecules identical to the parent. So you see that every time a cell arises, there are not going to be too many surprises because of this little template scheme, this biological Xerox machine. So when human parents make their contribution to a baby-to-be's constitution, since their chromosomes are human, we presume it's no shock that the baby is too. Well, I guess that brings us to the end of our tune, which began with a Martian in the waiting room. If you have any questions about something you missed, just send me an email. All class dismissed. I got to improvise a little bit.
I guess the last sentence was new. Send me an email.
Seiko, you're a real fox.
Come see me after class or something.
Well, in our business, we've successfully uncoupled travel from vacation. And this is a song called Take Me Home, TWA. Almost heaven, San Francisco, Mount Parnassus, striving arboretum. Life is fine there, the bridges and the trees. Elegant Victorians, always a joy to see. TWA, take me home to the place I belong. San Francisco, sourdough mama, take me home. TWA, all my memories of planes that went down, gents and ladies, strangers to each other, smoke and flames falling from the sky, drowning fears in moonshine, movies in our eyes. United Air, take me home to the place I belong. San Francisco, sourdough mama, take me home. United Air. I got a call in the morning at my hotel. Sound asleep, I thought I'd finally get some rest. The urgent news was that our sink clogged up again last night, and it had overflowed to the floor below. US Air, take me home to the place I belong. San Francisco, sourdough mama, take me home. US Air. I went to the airport for my flight, which was at 8 a.m. Half days I took a smelly taxi right out to O'Hare. Arriving at the gate, a sinking feeling came upon me. It was 5 till 8, time to run. Take me home, WA, to the place I belong. San Francisco, sourdough mama, take me home. TWA. I talked all day to 10 people, each for half an hour. They told me about their research while I tried to stay awake. My eyes, they kept on closing as I thought about the talk that I had yet to give. But would I live? US Air, take me home to the place I belong. San Francisco, sourdough mama, take me home. US Air. When I did dinner at the cafe pastoral across town, few gin and tonics, and I wasn't feeling any pain. The food was good, the talk was fun, but once again I knew that I just ate too much, just like at lunch. United Air, take me home to the place I belong. San Francisco, sourdough mama, take me home.